So thank you so much. I think Pankaj set the stage. I wish I had seen Pankaj's slides earlier. And Sagar gave a very nice insight. I'm going to be pretty rapid. I'm mindful of the time. I have 20 minutes. Systematic reviews and meta-analysis. So when you look at type of reviews, we have literature reviews, we have systematic reviews, we have meta-analysis, and you can see that this Venn diagram shrinks. And most anecdotally, people wrote as experts or so-called experts into inverted commas narrative reviews, which use informal or subjective methods to collect and interpret information. And these were narrative summaries of evidence. But when we really look at what's a way a systematic review is really collated, we have in vitro research, animal research, ideas, hypotheses, editorials, and opinions, case reports, case series, case control series, cohorts. And I was talking to Dr. Shah yesterday that we need to build long-term cohorts for our country. And we saw some very elegant collation of data from Sagar and his team. Congratulations to you and keep doing the good work you're doing for United States. And we need to do that in India too. And then we have these randomized, controlled, double-blind, tested trials. And then comes systematic review and meta-analysis. So from that standpoint, a review of an evidence on a clearly formulated question that uses systematic and explicit methods to identify, select, and critically appraise relevant to the primary research, and then extract and analyze that data from the studies that are included in the review. So that's how it should come in. So clearly, between systematic and narrative, the systematic approach is to review the article, criteria determined at the outset, comprehensive search of relevant articles, methods of appraisal and synthesis explicit, and meta-analysis is a part of it, can be used to combine data. Well, of course, narrative is very biased. It's very didactical and anecdotal. Author can pick up any criteria. It could be arbitrary at times because we live in an evidence-based era. The methods are usually not specified. Views could have a political tinge to it. Vote or count or narrative summary. And you can't really replicate such a review in evidence-based world. So clearly the systematic review has to reduce bias. It has to be replicable or reproducible. It should resolve controversies surrounding conflicting studies. It should identify gaps in current research. And it should provide a construct or a reliable basis for decision making. Because ultimately, our providers, we are here for our patients. Also, the limitations of systematic reviews are sometimes the reviews tend to be very inconclusive. There may not be enough evidence or trials. The trials may be of poor quality. The intervention may be very complex to be tested by a trial. And often, practice does not change just because you have the evidence of effect or effectiveness. So when we are writing up a systematic review, we need to formulate a question, plan a review, have a comprehensive search. We have very good engines for that. Have an unbiased selection and abstraction process, critical appraisal of data, synthesis of the data, and that includes meta-analysis. So that's the last component of my talk. Interpretation of results, and then reporting the review. So asking an answerable question is the key when you start a review. And the review questions are, on a treatment and therapy, which treatment is more effective? Does it do better or than harm? An example is hydrocolloid occlusive dressing better than a conventional gauze dressing to heal a chronic wound, say, in case of diabetes? Simple. Prevention. How to reduce the risk of the disease? Does increasing physical activity reduce the risk of developing diabetes? Diagnosis. How to select and interpret diagnostic tests? Is MRI more effective than X-ray of identifying a hairline fracture? Prognosis. How to anticipate the likely course of the disease? Are babies born who are bottle fed more likely to be obese in adulthood than babies who are born who are breastfed? Causation. What are the risk factors of developing a certain condition? Does exposure to smoking in mothers who smoke during pregnancy increase the risk of fetal death? So this is how review questions are often framed. And then we have the PICO, the patient or the problem, the P. intervention which is identified, the I, the C is the comparison, and the O is the outcome. So clearly when we write and think of a review, always go through the PICO filter, the patient or the problem, the intervention, the comparison, and the outcome. And usually, you know, you can have a PICO question. For example, a time-consuming question can be, what's the best strategy to prevent smoking in young people? An answerable question, and Dr. Shah works in public health, a mass media or a school-based or a community-based intervention is effective in preventing smoking on young people. So that can be an answerable question. So the problem population in the PICO T chart would be younger people under the age 25. The intervention could be TV, radio, newspaper, billboards, posters, leaflets, booklets. The comparison could be school-based or no intervention. The outcome measures could be the objective measures of smoking, like a saliva thiocyanate or alveolar or carbon monoxide, a self-reported smoking behavior, or the intent, attitude, knowledge, skill measure. These are intermediate matrices, media reach. And then, of course, we have the hard studies, RCTs, controlled before and after studies, or the time series constructs or designs. And then, of course, we can build research questions in different scenarios and how the components are related, say, in therapy, in prevention, in diagnosis, in prognosis, in etiology, or sometimes even experiment. So when we are writing a review protocol, we need to have three things: ensure transparency, ensure accountability, and avoid duplication. And in the objective section, have a single sentence derived from the research question, should relate to the PICO elements, in particular the population, intervention, and comparison, and stay very focused on the question. So when you're looking at a protocol in the study selection, whether two authors will independently assess studies, have a process of assessment of screening the abstract and then the full text, have a policy within the authors how disagreements will be managed, and will other methods, including software, used to select studies, because now we have to do a lot of data mining. And of course, data categories to be collected. Two authors will independently collect the data, piloting and using instructions for data collection forms, disagreement management, and clarification from study authors. So once you have a well-formulated question, you need to do a comprehensive data search. Then, of course, you have unbiased selection and abstraction process, critical appraisal, synthesis of data, and interpretation of results. So a good search should have a clear research question, should be all domain pervasive and comprehensive without language restriction, including unpublished and published research, because now there is a lot of unpublished peer-reviewed data also available. Often more than 60% of what is available in the global internet domain often doesn't even get into the final abstraction process. And of course, then look at the document research with replicability. We have a lot of core sources of published literature, like Cochrane reviews and Medline, the MBase, the Web of Science, the Campbell Systematic Reviews, the Scopus, the CTRIs and the SCADs and the InMeds. The Google Scholars are great literature, I found them. Open Gray sourcing is available now in Europe. We have the social SSRN, the ProQuest, and the Shodh Ganga from Indian University reservoirs, and so on and so forth. When you select studies, keep accurate records and tracks, have a good summary of the records you have searched, identify the excluders, provide reasons for the articles which are excluded, which are assessed in the full text, present a Prisma flow diagram, very, very crucial when you write a review, and keep the reports complete. Data types are, of course, dichotomous, continuous, ordinal, count and rates, and time of the event. And then try to grade recommendations. The grading of recommendations, assessment, development, and evaluations is a very transparent framework for developing and presenting summaries of evidence. Very low, low, moderate, or high. And I think it's very crucial. So clearly, when we look at, filtering information from background to case control and case series to cohorts to RCTs, we are moving the grid from unfiltered information to filtered information. And we are looking at a hierarchical increase in quality of evidence. And the TRIP database searches this simultaneously and we move from an unfiltered space to a filtered space. Therefore, including a Prisma statement of the identification of records which are searched, which we put in the review, the additional records, the duplicates which are removed, the record screen, the records excluded, the full text articles which are eligible and which are excluded, the studies included for qualitative synthesis, and then you put that meta-analysis. And that's how the whole Prisma statement is made. And then we move to a meta-analysis, which is the second part of my talk, for which I have around 9.5 minutes. It's a process. Meta-analysis is a very systematic review of quantitative estimation. So what we did before that was a filtering process of asking questions and putting it in a construct, while meta-analysis is a very, very quantitative estimation of the magnitude of effect and the uncertainty of that, because we want to look at a rigorous method eliminating bias so that we can get a confidence limit and put a matrix to it. So clearly, when we are conducting a meta-analysis, which is always a part of a systematic review, we need to formulate a research question, like defining a research question, defining inclusion and exclusion criteria, locating and selecting studies, extracting data, defining effect estimates, tabulation, data analysis and interpretation, testing homogeneity of studies, then investigating the sources of heterogeneity, because there's a lot of heterogeneity, and then trying to clean the data up, and then making it in a very presentable format based on the outcome of interest from the primary studies which are extracted from the results section. So doing a meta-analysis is easy, but doing well is a hard thing. The three most common effects of a dichotomous outcome are risk ratios, also often called in the meta-analytic terminology as relative risk. Second one, which we often use in research, is odds ratio. And third is risk difference. Sometimes popularly in popular scientific world, we call it absolute risk reduction. So those are the three variables which we often use. So risk ratio is nothing but to calculate risk ratio, take the risk in the intervention group divided by the risk of the control group, and the risk is calculated by dividing the number of events by the total number of people in the group, often done for the cardiovascular outcome trials and various other outcome trials. The odds ratio is odds of an event in an intervention group, which is divided by the odds of the control groups. And odds are calculated by dividing the number of events by the number of non-events. And the risk difference is nothing but absolute measure, giving you the absolute difference between risk of each group. So it assesses the events divided by total number of population in the intervention group and subtracts it from the risk of the control group. There is a lot of heterogeneity. What is heterogeneity? A lot of variation or differences across studies. Because when you are doing a systematic review with meta-analysis, every study will have a different design, different inclusion and exclusion criteria. So there will be a clinical heterogeneity, there will be a methodological heterogeneity, there will be a statistical heterogeneity. And that needs to be homogenized, and that's the biggest nightmare. And that's why if you want to make a good meta-analysis, that's the hard part of it. The good meta-analysis is the one which really harmonizes this heterogeneity, and I think that's really an art. So dealing with heterogeneity, when you plan a meta-analysis model for your protocol, look at studies which are dissimilar, and then don't proceed with your meta-analysis if it's very dissimilar. If it is not, and there is heterogeneity present, then present the overall analysis. But there is heterogeneity, then look at the data, explore the reasons as pre-planned, look at the found effect of modifiers, and then look at the subgroup analysis, and then look and cautiously discuss the effect of modifiers or outliers as people call them in lay literature, and present the overall analysis and also make a mention, notable mention of the unexplained heterogeneity. So to identify heterogeneity, we use the lab plot, a plot which shows the observational event rate in an experimental group plotted against the observed event rate in the control group for each study. And these lab plots are used with a range of event rates in trials to highlight the excessive heterogeneity. So sometimes this gets complex when you interpret it, but interpretation of this is very useful. Then we have the Galbraith plot, which is a plot of standardized intervention, which is an intervention of effect divided by the standard error against the reciprocal of the standard error, which is precision. And imprecise estimates of the effect lie near the origin and the precise estimate farther away. So they give you a correct impression of being more informative and accurate because of the inverse standard error or the z-score. And finally, we have the funnel plot. The funnel plot is nothing but a scatter plot of effect, which actually is a measure of precision or study size, and it mathematically plots the effect of estimates on standard errors. And usually these funnel plots are very good visual aid for picking up bias or heterogeneity based on the level of asymmetry in the data. So clearly when we present a heterogeneity in a presentation of a meta-analysis, this is a forest plot of a graphical presentation of summarizing all essential information of a meta-analysis. You can see the study, the experimental and the control, the mean difference, and the various parameters here. So also remember, bias reporting is very crucial. Bias reporting could have author beliefs, word restrictions, editorial approval, peer review, published reports, and so on and so forth. And therefore, reporting a systematic review or meta-analysis follows a simple list. Report the literature scoping containing combinations of search strings and the outcome of various databases. The list of articles excluded after reading the full text, with the reasons for exclusion. List of articles which could not be obtained on full text despite of efforts of the corresponding authors, of librarians, editors, Cochrane's, etc. Extraction of data and quality assessment, including Excel spreadsheets, because you know the supplementary tables can be huge, sometimes analyzing hundreds of pages of supplementary information. I'm sure Sagar made a very elegant mention of those CDC databases and various free databases. They're huge Excel speeds, and it's not easy how to include and exclude them in the studies. So when we look at this, working example, just a efficacy of a new molecule, for example, this is lobeglitazone. It's a systematic review and meta-analysis which we conducted because this molecule was a next generation of glitazone, and we had to conduct a meta-analysis and a systematic review. So we looked at, efficacy and safety of this novel compound with standard of care. We looked at all the databases for the relevant randomized controlled trials before it was launched in India. We looked at its primary outcome on comparison with A1C in 24 weeks. We looked at the pooled mean differences of the odds ratio and calculated the random effect models. We looked at almost 267 studies which were screened. Only 4 were included because the data was very less. And then we tried to use the grade model. And then we looked at a conclusion that treatment as an adjuvant with lobeglitazone changes glycemic status and lipid profile similar to standard of care. There was no statistical difference between this molecule and other which are already available with a reasonable safety profile. This was the Prisma plot which I told you. Look at how the database search started. 267, PubMed 57, Embase 137, Cochrane Central 40, Scopus 33, Clinical Trial GOV and MedReview, which is the unpublished version 00. Duplicates were removed. Record screened were 142 from 267. Full text articles for eligibility were only 11. And ultimately what we included in the review were only 4 for meta-analysis. So look at how the filtering process actually occurs. And therefore the Prisma diagram is very crucial. And this is all we looked at. And then again, they were only from those geographies where the molecule was available. Jin, Kim, Jin, Lim, that's it. And that's what was the data which was available in public domain, which we could then reproduce in a meta-analysis in a compound which was available in India, for which we were now going to use, and we have it available for maybe a year and a half. And that's how you need to recognize the power of current modeling methods to write a meta-analysis and a systematic review. This is how we wrote our discussion, and the full text is available. And for brevity of time, I'm going to stop here and just give you one last slide again, that systematic reviews and meta-analysis are at the top of the pyramid. Starting from in vitro and animal research, to ideas and hypotheses, to case reports and case series, to case control and cohorts, to RCTs. The true collection is a good meta-analysis and a systematic review. And meta-analysis is a part of a systematic review. And therefore, using this entire construct is very useful. I've been a little quick in my approach because of only 20 minutes given to me. Initially it was 30. Then Bansi, knowing Bansi fully well, made it 30 to 20. But these slides are available for each one of you for consumption, and if there's any guidance I can provide, I'll be happy to help. Thank you so much.